Assalamu alaikum. I am Dr. Khadiza, Associate Professor, Department of Physiology, Bashundhara Ad-din Medical College. Welcome you all in this online class. Dear students, how are you? Hope you are doing well. Today we are going to study a new topic, that is endocrine pancreas. This topics is item number 6. This includes the hormone of islets of Langerhans pancreas, its function, mechanism of action and regulation of secretion and disorders. Now you know that pancreas is a mixed gland. It is a leaf-like organ. It is found in, it is an abdominal organ. It lies behind the stomach, and it has body, head, neck and tail and an uncinate process. And histologically it has two types of cell, that is the exocrine portion, the acinar cell, and the islets of Langerhans, that is the endocrine portion. The exocrine portion, it makes about 80% of the gland and secretes digestive juices into the duodenum, and the endocrine portion it makes about 2% of the volume of the gland and secretes insulin and glucagon mainly, and it secretes secretion is into the blood, and then it transported through the blood to the whole body. The islets are scattered throughout the pancreas, although they are more plentiful in the tail than in the body and the head. The human pancreas has about 1 to 2 million of islets of Langerhans and organized around small capillaries into which its cell secretes their hormones. This is the figure showing the islets of Langerhans, and islets of Langerhans has 3 types of cell, that is alpha, beta and delta cells. Among them, beta cell is about 60% and it secretes insulin and amylin, and alpha cell is about 25%, it secretes glucagon, and delta cell, that is 10% and it secretes somatostatin, and another cell is PP cell, it is about 5% and secretes pancreatic polypeptide. Insulin inhibits glucagon secretion and amylin inhibits secretion of insulin, and somatostatin inhibits the secretion of both insulin and glucagon. Now we are going to discuss about insulin. It is the main topics. Insulin is the most important hormone controlling plasma glucose concentration. It is the principal anabolic hormone of the body, and it is called hormone of abundance. Now we are going to discuss the structure of insulin. Insulin is a polypeptide hormone. It is about 51 amino acids. It has 2 chains, mainly A chain, that is 21 amino acid, and B chain, this is 30 amino acid. It is linked together by 2 disulfide bridge, and human insulin has a molecular weight of about 5808. This is the structure of a insulin. Now we are going to discuss the biosynthesis of insulin. Insulin is first synthesized as a pre-proinsulin, then conversion of pre-proinsulin to proinsulin, and then conversion of proinsulin to insulin. How it occurs? Let us see. Insulin has a molecular weight of about 5808 and it is synthesized in the beta cell, and insulin is synthesized as a pre-proinsulin that has molecular weight of about 11,500 in the rough endoplasmic reticulum. Then it is then cleaved into endoplasmic reticulum to form a proinsulin, and that has a molecular weight of about 9,000 and consists of 3 chain, that is of polypeptide chain contain A, B and C. Most of the proinsulin is further cleaved in the Golgi apparatus to form insulin, composed of the A, B chain connected by a disulfide bond, and then C chain of peptide, that is called C peptide or connecting peptide. The insulin and the C peptide are packed in the secretory granules and secreted in the equimolar amount. It is about 5 to 10% of the final secreted product is still about in the form of proinsulin. Now we will discuss what is C peptide. C-peptide has no detectable biological activity. Plasma half-life of C-peptide is about 30 minutes. So, it provides a useful index in the rate of insulin secretion. Measurement of C-peptide levels by the radioimmunoassay can be used in the insulin treated patient, diabetic patient, to determine how much insulin of his own pancreas can produce. Store and release. Insulin is stored in the cytosolic granules. Upon appropriate stimulation, the secretory granules fuse with the plasma membrane and discharge its insulin into the extracellular fluid by the process of exocytosis. Now, we will discuss the metabolism of insulin. Insulin is secreted into the blood. It is circulated almost entirely in unbound form. Then it has a half-life of about average 6 minutes. It is mainly cleared off from the circulation within 10 to 15 minutes. It is degraded by the enzyme insulinase mainly in the liver, to a lesser extent by the kidney and the muscles. Now, what is the basic mechanism of insulin secretion by the beta cells of pancreas? That is glucose. Glucose is the main stimulus for secretion of insulin from the beta cells. Whenever the blood glucose level increases, it enters into the beta cell and it causes increased ATPase into the ATPase in the cell, and that causes inhibition of the potassium channel and opening of the calcium channel, and that triggers the entry of calcium into the cell, and that calcium causes exocytosis of insulin secretion to the exterior. In the figure you can see the mechanism. Now, we are going to discuss the mechanism of action of insulin. Insulin has already triggered by the increased blood glucose level into the circulation, and then this insulin will circulate throughout the blood and it has to act on the target tissue. So, what is the mechanism? You know, insulin is a protein hormone, so it binds to the receptors on the cell membrane. As it is a protein hormone, the receptor usually present on the cell membrane. Now, in the flow chart you are going to see the mechanism of insulin. Insulin first bind with the activation and activate the membrane receptors, and that causes subsequent effect. The insulin receptor is a combination of 4 subunits held together by a disulfide linkage. 2 alpha subunits that lies entirely outside the cell membrane and 2 beta subunits that protruded through the membrane and to the cytoplasm. Then the insulin bind with the alpha subunits on the outside of the cell and autophosphorylation of the beta subunit due to linkage with the beta alpha subunits. Then beta subunit activate tyrosine kinase and causes phosphorylation of multiple other intracellular enzymes, including a group called insulin receptor substrate. The net effect is to activate some of these enzymes while inactivating others. In this way, insulin produces the desired effect on carbohydrate, protein and fat metabolism. This is the figure showing that insulin bind to the receptors and activate the tyrosine kinase activity and activation of insulin signaling cascades, and the biological effects are insulin glucose transporter and causes protein synthesis, lipid synthesis, increase glycogen synthesis and growth and gene expressions. Now, we are going to discuss the major actions of insulin. Major action of insulin are anabolic and anti-catabolic action, anti-diabetogenic action or hypoglycemic effect, anti-lipolytic effect and anti-ketogenic action, stimulates potassium influx and growth regulation. Now, we are going to discuss the physiology effect of insulin that can be divided into rapid, intermediate and delayed action. Rapid action that begins within seconds, it increases transport of glucose, amino acid and potassium into the insulin sensitive cells. An intermediate action that is begins within minutes, and that is stimulation that stimulate protein synthesis, inhibition of protein degradation, and activation of glycolytic enzymes and glycogen synthesis, and inhibition of phosphorylase and gluconeogenic enzymes. And delayed action that begins within hours, that increase in messenger RNAs for lipogenic and other enzymes. Now, we are going to discuss the metabolic effect of insulin. First, we will discuss the carbohydrate effect on carbohydrate metabolism. Insulin increases glucose, increases uptake and use and storage of glucose by almost all tissues of the body, especially muscle, adipose tissue, liver, and thus decreases blood glucose level by increased glucose uptake, increased gluconeogenesis, increased glycolysis, increased decreased glycogenolysis, decreased gluconeogenesis, and increased lipogenesis from glucose, and all this together decreases blood glucose level. Now, what are the effect of insulin on fat metabolism? Insulin promote fat synthesis and storage. Insulin increases the utilization of glucose by most of the body's tissues, which almost automatically decreases the utilization of fat and thus functions as a fat sparer. That is, it increases lipogenesis, decrease lipolysis, decrease ketogenesis, and that last insulin increases fat storage in the adipose tissue. Insulin increases fat storage in the adipose tissue by insulin stimulates lipoprotein lipase, insulin inhibits the action of hormone sensitive lipase, insulin promotes glucose transport through the cell membrane into the fat cells. Now, effect of insulin on protein metabolism and on growth. Insulin promote protein synthesis and storage by increasing amino acid uptake into the cell, increase protein synthesis, decrease protein catabolism, and decrease gluconeogenesis. Insulin and growth hormone interact synergistically to promote growth. Effect of insulin on electrolyte metabolism. It enhances movement of potassium into the cell. Insulin increases the activity of sodium potassium ATPase in the cell membrane, so that potassium ion is pumped into the cell. There are insulin sensitive cell are mainly skeletal muscles, cardiac muscle, and adipose tissue. All these cells have GLUT4 transporter that is responsible for transport of glucose into the cell. This figure shows that insulin first bind to the receptors, and then it causes synthesis of transporter, that is GLUT4, that promote glucose to enter into the cell. So, if there is no insulin, that glucose will not enter into the cell, this cell, that is skeletal muscle, cardiac muscle, and adipose tissue. Now, we are going to see the effect of insulin over various tissues. First, on adipose tissue. On adipose tissue, it increases glucose entry into the cell, increases fatty acid synthesis, increase glycerol phosphate synthesis, increase triglyceride deposition, activation of lipoprotein lipase, inhibition of hormone sensitive lipase, increase potassium uptake. On muscle, it increases glucose entry, decrease glycogen synthesis, increase amino acid uptake, increase protein synthesis in the ribosome, decrease protein catabolism, decrease release of gluconeogenic amino acid, increase ketone uptake, and increase potassium uptake. And in case of liver, it decreases ketogenesis, increase protein synthesis, increase lipid synthesis, and decrease glucose output due to decreased gluconeogenesis, and increase glycogen synthesis, and increase glycolysis. And in general, it causes all cell growth. Now, we are going to know what are the insulin independent cells, that this cell does not need insulin to get entry into the cell. These are the brain, RBC, testes, kidney, retina and intestinal mucosal cells. So, students, that is all for today. We are going to discuss rest of the lecture in the next class. Thank you.